Okay, so I think over the next 30 minutes, we would like to talk about the development of children who were cooled for hypoxic ischemic encephalopathy. There's some of the questions I thought that would be helpful to address in this 30 minutes is, in infants cooled for HIE, what's the short to intermediate term outcomes? How do these children develop and their brain develops at school age? And often this question comes up. The next one, how is neonatal brain MRI associated with the short-term and school-age outcomes? And what do I tell parents of infants who underwent cooling for HIE with all this data we have regarding the prognosis or the outcomes? So the developmental outcomes which I would like to cover are about severe motor disability, which is cerebral palsy, borderline motor impairment, cognition, communication, attention, socio-emotional development, and behavior. So what we have been seeing is this improved outcomes in the hypothermia era. Hang on a second. Right. So this is the data summarizing death and disability at 2 years. On the left-hand side, the bar graphs are summarizing the data from the clinical trials of hypothermia versus normothermia. So the dark blue color shows the proportion of infants who died, and the diagonal lines indicate the proportion of infants who ended up with disability. So by 2010, hypothermia became the standard care, and then these are the data from the cohorts of children who underwent cooling. So what's quite striking is the mortality rate has come down, but if you look at the proportion of disability, that has remained the same. This is important because, yes, the practice of withdrawal of intensive care may have changed, but it didn't inflate the rate of disability. But why is that? If you look at that, one of the reasons could be, if you look at the data on the severity of encephalopathy of infants who were cooled in the clinical trials, there were, the dark red shows a severe encephalopathy infants, and the light red is moderate encephalopathy infants. The clinical trials, except the ICE trial, included infants with moderate to severe encephalopathy. And some of the trials, there are more severe encephalopathy infants than infants with moderate encephalopathy. But interestingly, if you look at the cohorts, if you look at the data on the distribution of severity of encephalopathy, it's predominantly infants with moderate encephalopathy who were cooled, and infants with severe encephalopathy seems to be fewer, and that might contribute to the reduced mortality rate. And also, you're seeing this gray shaded bar, which is showing that infants with mild encephalopathy are being cooled as well. So that's a therapeutic creep which is coming in now. So what are the measures of developmental outcome we use in clinical practice? Babies who underwent cooling, they undergo brain MRI in the neonatal period to help us with prognosis. We also do gentle movement assessment at discharge and at 14 to 16 weeks after birth to again help with the prognosis. Children undergo developmental assessment using Bailey scales of infant and toddler development at 2 years of age. And then that's the routine clinical practice. Beyond that, it's very rare children are followed up after having had HIE. So the school age development is quite patchy, and if at all, it may not happen at all in the NHS at the moment. So that's something which we are doing as a part of research. So the first outcome, cerebral palsy. What happens to these children in terms of having severe motor disability? So this data, the first 4 bar graphs are data from the clinical trials which happened before cooling became standard of care. And the last bar summarizes data from Bristol, which is a cohort study. Again, same, the dark blue, proportion of cooled infants who died, light blue, proportion of infants who survived with cerebral palsy. The numbers in red indicates a severity of CP. 1, milder CP, 5, severe form of CP. And in the clinical trials, we can see the severity of CP is uniformly distributed. You can see there about 4 to 5% had grade 1. The others had, grade 2 to grade 5. But what we're seeing in clinical practice consistently in different cohorts, they are predominantly milder CP. You can see 9% of children with mild CP and 5% of children with severe CP. Children with grade 2 to 4 CP, we do not seem to see. It doesn't seem to occur. But the key thing in this slide is about 70% of survivors do not have CP. So what are the outcomes of these children as they grow into school age? So this slide summarizes data on the school age outcomes from the clinical trials which are published to date. Here, the x-axis shows the different domains of intellectual outcomes, the full-scale IQ, verbal IQ, performance IQ, and processing speed. Here, the 100 is the mean, and the standard deviation is 15. So the average is about 100. The dark blue bar are data from United States NACHD trial, and the light blue bar are data from TOBI trial from the UK. It's very interesting that these data include children with and without CP. The data were obtained at 6 to 7 years of age. Children from the UK did far better than children from the US. I don't know the reason, but you can see their cognitive scores are better than the NACHD trial. But since cooling has become standard, what happens to these children at school age? That's the question which we had. So we did a cool MRI study where the case children are the children who underwent cooling, and these are children with HIE. They do not have any additional diagnosis. And we had age, sex, and socioeconomic status matched. Control children was at 43 children, and case children were 51. And if you look at the neonatal brain injury scores, about 20% had any basal ganglia thalami injury. This is, if you score, it's usually scored from 1 to 3, and none of them had grade 3, just 1 and 2. Because this cohort, we chose children who did not have CP because we wanted to understand how their brain develops. So we wanted to do advanced MRI on those children at 6 to 8 years of age. So at 18 months, if you look at the Bailey scores, they're all around the mean. So this is children who are at 2 years, you would say, normally developing. You don't need to be seen again. They are usually discharged in clinical practice. These are the early school age characteristics. They're about 6 to 8 years, and they're matched with a control group for sex and socioeconomic status as well. So these children underwent advanced imaging and developmental assessments using motor, cognition, and attention. So these are all clinician administered using MABC2 and Wechsler scales, Wechsler intelligence scales for children. And the attention is measured using a CPT test, which I'll go through. And we also assessed communication and social emotional outcomes using Strengths and Difficulties Questionnaire and CCC2. So neuroimaging, all these children underwent T1 and T2 and diffusion-weighted imaging. So we measured the volumes of the subcortical structures, the basal ganglia thalami and hippocampi, caudate and putamen, because these are the regions which are vulnerable to hypoxia in these children. We also looked at mammillary body structure. So these are this bulbous structure in the it's a brainstem nuclei posterior to hypothalamus, and this forms part of the Papez circuit, which is linked with hippocampus, fornix, and thalamus. And they are essential for emotional regulation and memory as well. And we use diffusion-weighted imaging. This is to show what metrics we used is in a simple metric, like a fractional anisotropy. What does it mean? It's a measure indirectly quantifying the white matter microstructure. I mean, these green circles are cross sections of the axons. If they are sparse or not myelinated, water will be freely moving in between them. So you will get a model, isotropy model like this, as a sphere. As the number of axons increase or they become myelinated, the water movement between them is restricted. And you can see the restriction in the water movement gives rise to this ellipsoid model. So it's a degree to which diffusion occurs in one direction in preference to others. That's what's a fractional anisotropy. And we also did some complex steps looking at the whole brain wiring, which is the whole brain connectivity. And then we looked at the strength of the whole brain connectivity between cases and controls. So going to the cognitive skills, all these slides will have the same color representation. The case children are shown by this orange color and the control children are shown by the gray color. To remind you, case children are the children who underwent cooling for HIE, and the control children are healthy children who are their peers and they're matched for age, sex, and socioeconomic status. It's very obvious here. Case children had a lower IQ scores. It's about 14 points lower than the control children. And if we look at the proportion of children whose IQ is less than 85, which is described as borderline to extremely low IQ, these are data from 3 different cohorts, beginning from 5 to 6 years to 11 years. And again, the cool children is about 13 to 25% of them as children grew, the proportion of children with difficulties increased as well with a lower IQ. If you look at the subdomains of the cognitive development, like perceptual reasoning, processing speed, verbal comprehension, and working memory, cool children scored lower than the control children. Okay, are there any underlying brain structural changes which can explain these findings? That was our next question. So we looked at the subcortical volumes, and here the control children are shown by the blue color and the case children are shown by a red color. The x-axis shows the subcortical structures. And what we found was that a thalamic volume, hippocampal volume, and putamen volume was reduced in case children compared to control children. And so was a global white matter and the gray matter volume. Okay, then we looked at how does it associate with their functional outcome, their IQ. So here on the left-hand side, the red colored scatter plot are from the case children, the blue colored scatter plot are from the control children. What we saw was the full-scale IQ was positively associated with the volumes of thalamus and hippocampus. And this is adjusted for the age, sex, and the total brain volume, and that's why you have the residual in the x-axis. So the lower the volume of these structures, lower the IQ. So then we looked at the mammillary bodies, and you can see this is a normal mammillary body, which looks like a little grape here. And if it is a grape which is smaller in size, it is partial atrophy that's equivocal. And if you can't see anything, it's a flat membrane, then it is an abnormal mammillary body. Here we can see on the x-axis the cases and controls. The proportion of children with abnormal mammillary bodies, about 35% of cooled children had abnormal mammillary bodies. And there's, there's nobody in the control children who had abnormal mammillary bodies, and they are equivocal in about 20%. I said earlier that the mammillary bodies are form this Pape circuit. It starts from the hippocampus and goes to fornix and then connects to the mammillary body. And then the mammillary body, through another tract, connects to the thalami. So our question was, how does the abnormal mammillary body impact the hippocampal size? So here on the x-axis, we have 3 groups: the control children and the case children with normal or equivocal mammillary body, or case children with abnormal mammillary body. What we can see is case children with abnormal mammillary body, they also had smaller hippocampi. So that proves that the damage which these children had in hippocampi extends through the whole circuit. We also looked at further the mammillothalamic tract and stuff, and we found abnormalities in these children. All right, the structures are abnormal. How does it impact the children? So if you look at that, the same groupings as we saw in the previous graph, control children, case children with normal or equivocal, or case children with abnormal mammillary bodies. And the y-axis shows the different subscales of the cognitive outcomes. If you look here, the full scale IQ, children who had abnormal mammillary bodies in the case group had lower IQ. If they had even normal or equivocal mammillary bodies, they still had lower IQ. And for example, the perceptual reasoning, all these subscales were lower in children with abnormal mammillary bodies. So there's an association there. Moving on, we looked at the white matter tracts. If the white matter tracts, I talked about the white matter microstructure, the fractional anisotropy. So we're looking at the fractional anisotropy here, comparing between the case children and the controls. The green lines are the white matter tracts, the white matter wiring. This is an average, and anything which you're seeing in an yellow color to red color shows those regions have impaired white matter microstructure in the cases compared to the controls. For example, you can see here the corpus callosum is lighting up as yellow color, and here's the internal capsule as well, cingulum and inferior fronto-occipital fasciculus. They all had impaired microstructure in cases compared to controls. So then we looked at, I was telling you about the technique of doing it is a little bit complex, so I'm not going into it, but the bottom line is how strong the global connectivity, the wiring in the brain is. And one of the thing is about localized processing. That is the ability of the brain to process the information within densely interconnected brain regions. And there are two parameters, local efficiency and clustering coefficient. So the graphs you're seeing in the left-hand side, data from the case children, and the one from the right-hand side are the case data from the control children. So what we see here is there's a nice association between the network segregation measures and the IQ in the case children, but we did not see that in the control. I think what we have to understand here is children who had lower IQ, they were noted to have reduced localized processing capabilities or reduced connectivity, global connectivity strength. And then the other measure is network integration. How does the brain process information from different regions which are distributed throughout the brain? That's integrating information. And there are two measures which we used. And again, same representation of data. Here's case children, control children. And again, children with, in the case children, you saw a nice association. The children with lower IQ, they had lower measures on network integration. So that shows there is some explanation in the brain architecture which could account for this low IQ in these children. Moving on to the motor skills, we used MABC2, and there are 3 domains: the manual dexterity, aiming and catching and balance, and the same color representation, but these are 3 cohorts. This is from Southampton, this is a Bristol cohort, the right-hand side is a Swedish cohort. And what we saw was children who were cooled, they had reduced manual dexterity skills and balance skills. And that was present at 5 to 6 years. We saw in Bristol, there was reduction in the manual dexterity. There was no control group in the Swedish data, but the pattern of scores remained the same. If not, it goes lower at 11 years. And we can look at if you look at the percentiles of these scores, if they are less than 15th percentile, they're often described as having borderline motor impairment. Again, the x-axis, we have 3 cohorts. The case children are compared to controls. About 20 to 30% of children have borderline motor impairment. Remember, these are children without cerebral palsy. They are going to mainstream schools. So these children continue to have borderline motor problems. And if you look at this, the evolution of this problem, this dark line is the borderline motor impairment, the dark black line, and the x-axis shows the age. So at 11 years, they are here. This is their motor development is really low. But then to start off with, by 2 years, they're doing okay. But as they grow, they are developing difficulties. So we then wanted to see, okay, we are doing Bailey scores, Bailey assessment. Can we actually pick up these children earlier? So the x-axis shows the Bailey 3 motor composite score, the threshold 85, which is 1 standard deviation below the mean. That's here. And then the y-axis shows the MABC total score. If they are less than 15th percentile, which is this top dotted line, then they could have borderline motor difficulty. This ellipsoid where I have highlighted, these are the children who scored fine on the Bailey scales at 2 years, but then these children evolved and developed difficulties by 6 to 8 years. So we were not able to pick them up early. It doesn't mean the Bailey scales are not good. It's probably these children are developing difficulties as they grow. And this is to show this is a control child who's threading into the blocks. You can see how well he's able to do. And we can compare a case child. So there are lots of things in this video. The attention, manual dexterity, and the executive function. All those things are a bit challenging for this child. So again, that we looked at, are there any underlying brain structural changes which could be associated with this motor scores? Here, the first 2 graphs are from the case children. The y-axis shows the total motor scores. X-axis shows the volumes of the hippocampus and the thalami. And these children, the ones with lower motor scores, also had lower volumes of the hippocampi and thalami. And these are the global brain connectivity strength measures, which we discussed before. We saw the similar pattern with motor scores as well. So the children in the case group with the blue dots, y-axis showing the total motor score, x-axis shows the 2 measures, local efficiency and the clustering coefficient. Children who had lower motor scores also had lower measures on their ability to do network segregation. The strength of the global connectivity is impacted. I mean, we looked at the network integration measures, and we looked at the association with the total motor scores and the network integration measures. Again, children with lower motor scores also had lower measures on their ability to integrate information from around the brain. We measured communication using children's communication checklist. These are questionnaires completed by parents. And here the case children in orange, when we looked at the global general communication composite score, the case children scored lower than control children. When we looked at the subscales, for example, the structural language score on the left, which measures speech, syntax, semantics, and coherence, case children did slightly, their scores were lower than the controls. When we looked at the pragmatic language scores, which measures aspects like inappropriate initiation, stereotype language, nonverbal communication, again, case children scored lower than control children. We did not see any difference in the autistic type behaviors between cases and control children. And when we looked at are there any association between the brain white matter integrity and communication scores, we found that certain regions of the brain, such as forceps major, forceps minor, and external capsule, those regions are associated with the global communication scores in case children, meaning children with impaired communication scores also had impaired white matter microstructure on in these regions. We also measured attention using Corner's communication performance test. In this, children are given a laptop and they have to press a mouse every time they see a letter other than the letter X. And this measures their ability to be attentive. For example, the first two measures, which is hit reaction time percentile, the higher the score, the higher their inattention is. And here you can see the case children denoted by the orange, they're all clustered at the top, suggesting that these children are highly inattentive. And the last measure, hit reaction time by blockchain, measures sustained attention. And again, the higher the score, they are not able to maintain attention for a long time. And there's a tendency, it's not significant here, there's a tendency that case children may not be able to have a focus attention for a long time. And we also notice in their brain imaging, the attention network, which is a superior parietal gyrus and postcingulate cortex, in those nerve bundles, the connectivity is reduced in cases than controls. Other groups have shown this as well. If you see here in this graph, the x-axis, teachers were asked to rate the attention score for these children, and the y-axis shows a total motor score. The dark blue circles are from case children. You can see the case children are scored higher for inattention. We looked at the executive function. There are 3 different measures. This is published by Southampton group, which is using the brief. So behavioral regulation index, metacognition index, global executive composite score. That shows this case children are scoring much worse in the executive function. They have deficits in their executive function. We asked parents to score their socio-emotional function and also the behavioral problems. And here, the case children shown in the orange color, they had higher emotional problems and conduct problems compared to control children. Okay, so these are the problems the difficulties these children may have at 6 to 8 years. So one of the questions often comes up is, okay, the children have a normal neonatal MRI, will they be normal as they grow up? So this is data from Netherlands and Switzerland as well. So here, what they've done is there are different. We can discuss this. The clinical MRI which we do is not the same as research MRIs. The research MRIs are thin slices, and again, it's a qualitative scoring. It depends on the expertise of the neuroradiologist as well in terms of the scoring them. So here is a score which was used was a gray matter score, which is looking at the, I showed you the deep brain regions which are vulnerable to injury. They scored. And this is the cohort 1, which is the Holland, and this is cohort 2. And this is the outcome at 2 years of age. Children with normal outcome, and then they compared their gray matter score. Children with abnormal outcome compare their gray matter score. So what they're showing here is if they have a threshold of 9.5 at 2 years, and then majority of the children with abnormal outcomes had a score above that, whereas at 6 to 7 years, I think they had up to 8 years, they had, if the score is above 11.5, then they had abnormal outcome. But you have to be careful how these abnormal outcomes are defined in the clinical, in the research versus what happens in the clinical practice. This is a composite outcome: death, or children with CP, or children who had scores less than 1 standard deviation below the mean in the Baileys or the IQ. But the number of children, if you see, there's only 4 children with Bailey less than 85, or 3 to 4 children with IQ less than 85. So it is actually predicting children with severely abnormal outcome, death or CP, really well. And that's shown consistently in different studies. Because again, if you see here, there is differences between this cohort and this cohort. Children with abnormal outcome, they still had normal gray matter score at 2 years. And children with abnormal outcome still had normal gray matter scores at 6 years. When we looked at the Bristol cohort, it's using a different scoring method. So this is neonatal MRI score, which goes from 0 to 11. And the y-axis is the Bailey cognitive score. I told you 85 is a kind of threshold, 1 standard deviation below the mean. The lower the number, better the brain imaging, no injury to worst injury. You can see, yes, as the injury scores increase, you can see children Bailey scores were lower. But there are also children who had lower Bailey scores when they are, having lower imaging scores. The key thing is this slide. If you look at the x-axis, this shows the deep gray matter injury score, BGT predominantly, and we combine with the white matter score as well. And all those triangles are children with cerebral palsy. There's different grades of cerebral palsy: the red, green, and the orange, and the circle as well. And these stars are children who died. So if you look at this, as the MRI scores worsened, you can see it picked up more children with CP and death. So MRI deep gray matter injury is really sensitive in picking up children who might develop CP later on. Although you may ask, there are, none of the test is 100% foolproof. There will be some children who we may not be able to pick up, and these are these, you can see 2 or 3 children who the scores were not higher on the MRI. So then we looked at the neonatal MRI score. This is on the y-axis here, and then at 6 to 8 years, their IQ. So 0 means no brain injury. And you can see the difficulty here. You have 0, but then you have about, these are binned scatter plot. So as the color intensity increases, the number of children in that circle is higher. So for example, no brain injury, you still have about 4 to 5 children with IQ scores about 80 to 84. And then you have children with higher injury score of 6, they are scoring IQ about 110, 105. So it is a challenge when it comes to an individual child. For population level, it might be good in predicting composite outcomes. And we've looked at the relation between the Bailey and done at 18 to 21 months and their VISC IQ done at 6 to 7 years in these children. So the x-axis, you can see, these are the cognitive scores. 100 is the mean in the population. The blue line, you can see this distribution of the Bailey scores for these children. So the mean is about 103. And when we followed the same children at 6 to 8 years, you can see this green line, which is shifted to the left, and that's their IQ. So the IQ is 94.9. So by the time they grow into school age, their cognition, because that's when the cognition develops. You can't pick up reliably cognition at 18 to 21 months. So the cognitive skills evolve after 2 to 3 years of age. And you can see the cognition is much lower at the time. I'll leave you with this excerpt from a parent's email to read what challenges these children face at school.